Welcome to the Great Lakes HABs Collaboratory and Invasive Mussel Collaborative webinar, Exploration of HABs and Invasive Mussel Interactions. We apologize for the slight delay due to some technical difficulties, but we think we're all ready to go now. I'm Mary Ann Evans with the U.S. Geological Survey.
And I'm Erica Jensen with the Great Lakes Commission.
On behalf of the HABs Collaboratory and Invasive Mussel Collaborative, thank you for participating in today's webinar. For those of you who are new to one group or the other, the Great Lakes HABs Collaboratory was formed in 2015 with a mission to create a collective virtual laboratory that enables science-based information sharing and collective action to address HABs in the Great Lakes. Our webinar series was developed to improve communication and knowledge transfer and to create new opportunities for collaboration by presenting brief overviews of recent, ongoing, and new research projects. The Invasive Mussel Collaborative is identifying opportunities to advance scientifically sound technology for control of dreissenid mussels in order to produce measurable ecological and economic benefits. The collaborative provides a framework for communication and coordination and is working to identify the needs and objectives of resource managers, prioritize supporting science, and align science and management goals into a common agenda. If you would like to know more about either group, we invite you to visit our websites at the addresses on the screen. The websites are intended to serve as a central hub of information. Each also includes background information on the collaboratives and recordings of past webinars. If you know of information or have resources that you would like to see added to either website, please contact us. We also encourage you to join our respective listservs if you've not done so already, to receive webinar announcements, read about recent dreissenid or HABs-related news, and to connect with other researchers and managers. A link to join the Invasive Mussel Collaborative listserv is provided on our website, and you may join the HABs Collaboratory listserv by emailing Ken Gibbons at KGIBBNS. Sorry about the back there. Does one of the presenters have their phone on speakerphone?
Just switch it to mute for the time being, please.
Thank you. K. Gibbons at glc.org. Today's webinar is a joint webinar for the purpose of facilitating learning and information sharing on HABs, dreissenid mussel relationships. Several interactions have been hypothesized between HABs species and dreissenids through nutrient cycling and the nearshore shunt, through selective feeding changing algal species composition, through food supply to mussels, and through other mechanisms. We are pleased to have representatives from Wayne State University, the National Oceanic and Atmospheric Administration, and the U.S. Geological Survey to share their experience and recent research covering various theorized HABs dreissenid mussel relationships. Today's webinar will be recorded and archived along with past and future webinars on both collaborative websites. In just a few minutes, we'll have our first presentation. Before we begin, I have just a few logistical items to cover. We are pleased to have more than 275 people registered for this webinar. Due to the large number of people participating, all attendees' lines are on mute. Attendees will remain muted throughout the webinar, and we can only be taking questions in written form. If you have a question, you may type it into the questions pane on your GoToWebinar dashboard. Questions may be submitted anytime during the webinar. Depending on time, we may answer one or two questions following each presentation, but the majority of questions will be addressed after all 3 presentations have been given. We will do our best to take all questions or comments during the webinar. If you have to leave the webinar before your question has been addressed, we encourage you to review the webinar recording that will be posted on our website to hear your question and answer. If you are experiencing technical difficulties, you may send a chat directly to Sarah Cook, listed as an organizer for this webinar. I will now turn things over to Erica, who will introduce our speakers.
Thanks, Marianne. And thanks, everybody, again, for your patience this morning. We're ready to get started with our presentations. Our first presenter today is Dr. Donna Kashian. Dr. Kashian is a visiting scientist with NOAA's Great Lakes Environmental Research Laboratory and is a professor at Wayne State University. Her research focuses on the impacts of disturbance on water quality, encompassing both natural disturbances such as algal toxins and sedimentation, as well as those caused by humans, including contaminants, climate change, and introduced species. Centered largely in the urban center of Detroit, Michigan, her lab's research has been implemented into community-based educational programs to better inform the public of environmental health threats associated with many water quality disturbances. Thanks for joining us this morning, Donna. You should be ready to go. Donna, you may have to unmute your phone if you were on mute previously. We can't hear you yet.
Hi, thank you for that. Today I'm pretty excited to present the research I'm presenting today because we do, the work in our lab does very much work at the interface of both harmful algal blooms and invasive species, in particular dreissenid mussels. And the work being presented today is in large part also being conducted by one of my graduate students and a bag of holes. So what we're looking for is that interaction, that physiological interaction between dreissenid mussels and reproduction. There we go. So in looking at that, there is a lot we know about dreissenids and their interactions with cyanobacteria. So there's a whole bunch we know, but surprisingly, there's still a lot we don't know. And with dreissenids, we know that their impact is a lot because they're very prolific filter feeders, that they impact the phytoplankton community. So we'll see changes in phytoplankton dynamics, changes in species abundances, different species. And with that, we see the occurrence of also more cyanobacteria in the lakes. And Hank, who is the next speaker following my talk, was one of the people that initially identified that dreissenids can actually enhance cyanobacteria blooms through selective feeding. So they'll filter in the algae they want to eat, the healthy green algae, and so they'll consume that. But when the Mycocystis, in particular with his research, they consume that, they spit it back out and allow it to proliferate in a greater abundance than those green algae that have been consumed. So this in turn facilitates blooms. And although it's not an interaction, but it's important to note for this, this project that we also see in cyanobacteria a lot of different species producing harmful toxins, and of those species, different cultures of them, different strains, some of them will produce the toxins and some won't. And I think everyone on this webinar understands the impacts of recently, particularly in the Great Lakes, of these blooms causing lots of problems. And one of the most famous ones is being the shutdown of the water in Toledo following concentrations of mycosystem at high levels. So kind of to make sure we're all really we understand in doing this research that we have dreissenids persisting with cyanobacteria blooms. So we know that. So we're not, when we're looking at the impacts of cyanobacteria on dreissenids, we understand that they occur together, and cyanobacteria blooms haven't prevented dreissenids from proliferating in lakes. So that's a component we recognize, but we're trying to get at, does it have negative impacts? And if it does, then there may be some components of the physiology, the toxin, or the pheromone that can be used to actually be aligned with different control methods to make control methods out there more effective. So by continuous exposure of Mycocystis, for example, toxins may impair mussels. So in looking at the life history and physiology of dreissenids, we know they're broadcast spawners. So they release their sperm and eggs into the water. And in general, they spawn in the early summer. But what we've seen with the change in temperatures in the Great Lakes, we have actually seen spawning increase from very early April until September. So now we're starting, before we had these spawning events pre-bloom formation, but now we're starting to see spawning events happen during when we have August, September blooms. So we're starting to see this overlap. Like Winnipeg, we had that, and Lake Erie, we've had this happen. So this overlap is now occurring when it historically was not such an issue. We do know from the marine literature that certain mussel species rely on signals, pheromones, from phytoplankton to spawn. So the mussels, when the algae is a very good, healthy algae, it lets off a pheromone, and then the mussels know to spawn and release their veligers, release their eggs and sperm into conditions that are very favorable for their success. We don't know, it's never been identified yet in the Great Lakes if this happens. And it's not so much that we haven't found it, it really isn't something that's been looked for either from what we understand. So if indeed mussels can induce spawning, it is possible that cyanobacteria and other things out there may also release pheromones that say this is not favorable conditions to spawn. So we're also looking at focusing for this talk primarily on inhibitors. So do cyanobacteria release a chemical that inhibits spawning in some way? So ultimately, that is what we're looking at, determine the effects of cyanobacteria on dreissenid reproduction. And we're going to focus on spawning, fertilization, and sperm vitality. So for this, we collected our mussels from the Detroit River. Most of these mussels are quagga mussels at this point in time. And we tested 16 cyanobacteria cultures. Of those, there were 8 species. And the reason we did this is because, as I mentioned earlier, I think most of us do recognize that certain species, especially like Mycocystis, some strains will produce a toxin and some won't. So we wanted to look at a variety of species, especially in the ones that are really common for blooms in the Great Lakes. So for Mycocystis, we got one of our strains from Tim Davis at Glural, Noah Glural. We got, and that one was from Lake Erie. We have a strain from Muskegon. We have strains from Sue Watson in Canada. And also purchased strains from like the University of Texas algal repository. So we have a pretty good variety, so we can really look at the various effects. So I know this slide's a little bit busy, but it has all 3 methodologies listed out. We looked at spawning assessment, and this was primarily done by using serotonin, which is something that we know induces spawning. So you would take the control mussels, expose them to serotonin, and also expose the treatment ones, which were also exposed to the cyanobacteria, and compare cyanobacteria mussels to the control mussels and see which of the mussels are spawning, both males and females. For fertilization, these were done separate than the spawning assessments. We actually induced spawning not in the presence of cyanobacteria. Then we collected the sperm and eggs and placed them in well plates, at that point exposing them to our 13 different strains of cyanobacteria, and looking to see if fertilization occurred. And we were looking for mitosis and cellular cleavage at that point, which we were able to identify and actually rear some of the veligers up. And then finally, we also looked independently at sperm vitality. And so once we induced spawning, we were able to collect the sperm, put them in well plates, and put them under a very high power lens and use image tracking software to measure various components of movement, velocity, acceleration, and things like that. So we're trying to hit that reproduction and try and figure out what particular mechanism, if there is an impact, that it is on. So just briefly looking at the results, here we have one of the algae that we saw results from, Aphanizomenon. And looking at the control, which are the black dots on the x-axis, down here we have chlorophyll A concentrations, and that was just a standardized measure we had to measure how much of the the treatment algae we had. So this 0.0143 would be our lower concentration, and 0.607 would be our higher concentration of chlorophyll. And what you can see here in percent spawned on this axis. And so we have the difference between the algae, cyanobacteria exposed dreissenids, and then the controls. And when the concentrations got higher, you can see that we had spawning reduced by this particular cyanobacteria, which I am not going to pronounce again. So we also looked again at fertilization, and here on this chart graph, we did put most of our algae on here, and the asterisks represent where we had significant differences, and I'll put red circles around those too. So here we're looking at, again, the difference between the control, which is in the gray line, and the green, which is the particular cyanobacteria we're looking at, and looking at the difference between the control and its corresponding treatment of percent eggs. So the eggs and sperm were separated and put together. And what you'll see when you look at this, what's very consistent is everything that's significant is always in the same direction. It's always the cyanobacteria did reduce fertilization rate, percent fertilization of the mussel, sperm, and egg in these experiments. Likewise, looking at sperm vitality, and just in particular this one measure, you'll see the same thing. We have, I'll make the significant one stand out, we have the cyanobacteria and the control algae, the difference between this time blue and gray bars. And here again, in the several species, in this case, we had 2 species of microcystis, so an impact on the algae. And finally, with sperm acceleration, we see the same thing. I'm going to highlight the significance here. Again, with a microcystis species, we saw a difference, a change in between the control and the cyanobacterium acceleration. So we're having very consistent results when in all 3 different reproductive mechanisms. And it's not to say that, and to some extent, that these are not related between each other. So in summary, for fertilization, we had 7 cultures that that cyanobacteria had effect on decreasing fertilization. For spawning, we saw it with, in particular, 1 species, and with sperm motility, again, we saw this with microcystis and a famine Amazon also. And then we saw some differences between the culture, all consistent with decreases in motility, spawning, and fertilization. So we do know that now cyanobacteria can impact spawning and fertilization based on these laboratory studies. And so with this, why this information is useful, 1, we're getting a better understanding of physiology and potential impacts on life history of the dreissenid mussels, but we can also use this to enhance population mussel estimates for the spread of mussels and when they're spawning and the success of the spawning during different times of the years. And ultimately, what started this project, funding through USGS and ultimately through EPA, was developing a chemical tool for reducing reproduction might be derived from the cyanobacteria. I do want to note this is not a toxin causing this. So I think that was the first thing we wanted to rule out. So all of these assays were also looked at using microcystin, the pure toxin, and it never had any of the effects we're seeing when we use the algae. So now what we need to do, and our next step, is to identify what is it about the algae, the cyanobacteria, causing these changes. And we do think it's, if you notice, the ones that caused the effect were some of the cyanobacteria. They all had the mucilage sheath around it. And so we think it may be a polysaccharide associated with that colony-forming component of the cyanobacteria. We had a lot of people helping with us on this. USGS funded it. Once we started dealing with sperm and mobility, we had to go to the medical school. They were more equipped to measure sperm than we were in ecology. So we very much thankful for the groups that helped us. And thank you.
Thank you, Donna. So if folks have questions for Donna, we encourage you to type those into the questions box of the GoToWebinar dashboard. But at this time, we're going to go ahead and move to our next speaker, and we'll collect those questions and go through them after all of our speakers have presented. So our next presenter is Dr. Hank Vanderploeg. Dr. Vanderploeg is a research ecologist and ecosystem dynamics branch chief at NOAA's Great Lakes Environmental Research Laboratory, where he has spent most of his career studying the lower food web, invasive species, and spatial interactions of the food web. His research interests include food web interactions, particularly feeding mechanisms and life cycle strategies of plankton and benthos, and spatial distribution and interactions of all food web components. Hank, you are all set, but if you could, do you have two screens on your computer there?
Yes, you should be looking at screen 2.
Can you change it to the other screen in your settings? Because right now we're seeing your presenter view of your slides.
Okay, that's too, okay. Yeah, I'm not sure I can do that. Okay, yeah. Here.
Or we can just see your presenter.
Okay, there we are. Okay. Can you see
Perfect.
that?
Very good.
Thank you very much.
Okay. Yeah, I'll be talking about the mussel HAB collaboration. We'll be talking about the biophysical, also be touching on nutrient interactions because we have some recent publication on this that may be relevant, and how all these things interact to produce microcystis dominants, or in some cases, blooms. In this first slide, you can see interactions of mussels, and just to remind people, ceston contains a whole bunch of stuff. The mussels take it in. Stuff they don't like, they spit out as pseudofeces. If they do like it, it gets ingested and incorporated in their biomass, and it passes through the digestive tract and would be ejected as feces. They also produce nutrients that go back to the water column, which in turn may promote further phytoplankton growth. Now, this is what I call the dreissenid mussel wildcard questions. And we have some worries around dreissenid. As the mussels spread through the U.S., will microcystis blooms follow? This is an issue for some folks. The other is closer to home on Lake Erie. If we achieve a 40% reduction goal in phosphorus loading in Lake Erie, will HABs go away as predicted or hoped, or will mussels mess up this story? So the scientific question is, under what conditions will mussels promote microcystis dominance? This slide shows we've used a number of approaches to try to get at this question. We've looked at paired feeding and nutrient excretion experiments. I've worked with colleagues at Michigan State on enclosure studies. I've worked with folks on genetic characterization. And the other thing unique to a lot of glottal studies is to actually look at the behavior of what's going on. I'm just going to move through the story here. Our first work focused on selective rejection mechanisms in Saginaw Bay and Lake Erie, and our study sites were particularly Saginaw Bay, as you see here, as well as western Lake Erie. In these early experiments, we did very, very simple experiments. We measured changes in size fraction, chlorophyll in less than and greater than 53 micron fractions of chlorophyll. The larger fraction usually contained microcystis if present. And you can see we have a number of buckets, control buckets without mussels and buckets with mussels. We looked at concentrations of algae at the beginning, in the water column at the end of the experiment, and then we actually mixed up the whole containers to see what might have settled out. And this just shows a picture of the mussels in their baskets, which were added to the containers. Very, very simple in concept. The experiments were paired with videotapes of mussel behavior, and this was back in 1996. We did a few of these experiments, and we were ready for showtime as blooms really became a concern on Saginaw Bay. Microcystis blooms were seen on Saginaw Bay, and very large blooms. The water quality modelers said, hey, this can't happen. The nutrient concentration is too low for this to occur. I blamed the mussels and showed a videotape of what mussels do. This goes through very quickly. I hope you can see this. But you look at this mussel in the foreground, it sucks in a bunch of material here. These are microcystis colonies in the background. After collecting this stuff for a minute, they spit it out. And out of this rose the selective rejection paradigm. Large toxic colonies are rejected, while small algae, presumably nutritious species, are ingested. And looking at the original paradigm details, you need a lot of mussels that clear a significant fraction of the water column per day. The large microcystis are rejected. The pseudofeces, none of this gets ingested. The stuff comes in and it's spit out. And these are very loosely aggregated pseudofeces with microcystis returned to the water column unharmed. And at this time, we thought nutrients from processed algae returned to the water column to fertilize microcystis. At the same time we did these studies, we also had a large monitoring program on Saginaw Bay, and we did see that after before the mussels, there was a spring bloom and not much happening, but after mussel introduction, there tend to be a peak of chlorophyll in the summer, and you can see here from the slide showing 1992 and 96, there were an abundance of microcystis and other blue-greens, such as the Phaneropsa. I've already gone through this, but just to refresh your memory, again, stuff came in as ceston. Microcystis was rejected as pseudofeces after a short time of collection, and at the same time we presume nutrient excretion was going on. At the time, this was all very controversial. In fact, if we didn't have the films, I don't think anyone would have believed this, because people had observed that many laboratory strains of microcystis are filtered at a high rate. In fact, in our study published in 2001, we showed the same thing. We've seen no microcystis increases in mussel-invaded hypereutrophic lakes in the Netherlands. Cultures isolated from Dutch lakes and microcystis were readily ingested. We also saw in the Hudson River, at least the small colonies of microcystis were readily ingested. I might mention the large colonies were not in a study published later. So this has a number of forecasting implications. The idea there might be a nutrient trophic gradient in the interactions, and associated with this, there may be a nutrient interaction in terms of the nutrient stoichiometry story, ratios of nitrogen and phosphorus excreted. Strains may vary among lakes with different eutrophy, and the grazing may not be important at high phosphorus concentrations. If algae are growing so fast and there aren't a lot of mussels, then the mussels have no control of the situation. At the same time, our colleagues at Michigan State, led by Orlando Sarnelli, observed that as mussels spread into inland lakes in Michigan, there was an increase in microcystis in the low phosphorus lakes, but not in high phosphorus lakes. At the same time, not only was there a shift to dominance of microcystis, but there was also an increased toxicity in one of the studies not shown here. So there are a lot of questions floating around, and what did we do? We looked at the strain question, what factors induce rejection. We did more work with natural ceston using different methods of observing changes, and we looked at the potential effect of nutrient excretion of mussels on promotion of microcystis. And I'll just speak to, in the next slides, of the importance of working with freshly isolated cultures of natural ceston. Once you bring these things in the lab for a while, they're not the same wild creatures they were before. I'm going to show, each of these next slides you'll see our work with different cultures isolated by Alan Wilson. You see a little video clip. You see the concentration of chlorophyll in initial concentration at different size fractions. Again, greater than 53. These are generally large colonies with several hundred cells. The microcystin content, and you can see here the clearance rates. And in this particular alga, relatively high microcystin concentration, and they had relatively high clearance rate on the small size fraction. And I'll show you the video clip on that. You can see a cell or two went in, and after a few minutes, what happens is they've collected a bunch, and you saw a enthusiastic capture and forceful rejection. So they're pulling this in enthusiastically, but they are rejecting the larger microcystis and ingesting the smaller microcystis. Another strain we worked on, this is a very interesting one, very low microcystin concentration, but nothing was ingested. And I'm just going to show you a video clip with this Gilkey Lake strain. There really wasn't any feeding in any size category, and I'm just going to show you what an unhappy mussel looks like. Notice the siphon is not extended, and it's trying to get rid of this stuff, and it really can't. So this, if you had a pure culture of this in nature, nothing would happen. Now we added Cryptomonas. Cryptomonas is an ideal food for mussels. This is the same mussel. We did this with several mussels. After we did the video observations, we popped in some Cryptomonas, and we watched what happens. And you can see they're taking it in. But you saw that one colony come in and then was immediately rejected. That was a microcystis colony. As soon as it went in, it was rejected. It's very, very sensitive response. We saw mussels do this hundreds of times, so this isn't just an isolated interaction. We're going to talk a little bit about the mucilage. A lot of microcystis has a lot of mucilage around it, and this shows a picture of one of the strains with, you can see this halo of mucilage around it. And what you see here is overall there was no ingestion by the mussels. Microcystin concentration for chlorophyll was very, very low. You can see these guys enthusiastically captured stuff, and then every few minutes blasted away the stuff very forcefully. So Nothing else there. They were happy to capture it, but they rejected it. Later, we followed up by busting up these colonies into small pieces by sonication. And again, there was no ingestion. They just didn't like this non-toxic algae with lots of mucilage. Okay, so we had a number of conclusions. Colony size and mucilage is a sufficient condition for rejection. Microcystis, doesn't matter whether there were mussels in the lake or not, they were rejected. There is a toxicity response, and not necessarily microcystin, that makes the rejection process more sensitive. And there does not appear to be a clear connection between microcystin concentration and probability of rejection. Okay, so that's a pretty clear that they don't like a lot of microcystis strains or species found in lakes. Later on, we followed up by looking at more details on the phytoplankton. Later, by a Chinese visiting scientist, again doing these experiments in buckets. Doing the brute force algal counting, it took her a year to count through 5 experiments we did, and I'm just going to point out a few things. We can look down here at microcystis. It showed in this experiment they didn't like it, and this is something that pops up in, has increased in lakes where there are mussels. Aphanocapsa, another one they didn't like. This has popped up in a lot of systems. Anabina, they love this stuff. And Anabina is one of those algae that, one of those cyanobacteria that was a problem before the mussels came in. I'm just going to point out again, we're looking at Fragillaria, for example, another summer dominant, same size as microcystis. They really, really love this stuff. So you can look at these experiments with a number of species. They really like big stuff, the same size as microcystis, readily ingested it. So the point of all this is dreissenid can take out a number of algal species, big or small, and leave microcystis behind. Now I'm just going to visit the Gull Lake story really quickly. A. Sarnelli and his graduate students did experiments where they added different amounts of nutrients and mussels to the containers, and what was interesting is that in summer, when you add more phosphorus to the containers, and what happened is the enclosures added more phosphorus to it, you didn't see the microcystis dominance. It actually worked against the mussels, which tied in with the observation of mussels inducing microcystis in low phosphorus lakes, but not in high phosphorus lakes. Something that's impressed now, we've looked at the nutrient excretion interaction from actually studying mussels from these lakes, and phosphorus excretion is very sensitive to ingestion of phosphorus. If they're not feeding on algae, you're not getting any phosphorus excreted. So this is very important to summer dynamics of phytoplankton. On the other hand, you don't have to have much nitrogen in the system. They will excrete ammonia at very high rates. And there is a whole complicated nutrient stoichiometry story, but the bottom line is when you have high ceston and the P ratio, you have very low phosphorus excretion. Not true for ammonia excretion. So what this boils down to is the nutrient excretion that we've observed sort of runs counter to a lot of the ideas out there in the literature. Basically, phosphorus excretion will be low in a lot of these low nutrient lakes, and they will be also low when microcystis is dominant in summer where there's not a lot of feeding. Okay, at this point, I think I'll wrap up with this part of the study. Some things to consider here when, in terms of the mussels and microcystis. When there are a lot of mussels in the systems over a pretty broad range, mussel rejection of microcystis can be an important factor. The main thing is mussel feeding must exceed algal growth. The other important thing here is harmful algal blooms like hot conditions. Mussels do not like temperatures above 28 degrees for long periods of time. They get sick after a while. In fact, one year when we were doing these experiments, the mussels all died on us because it got too warm in the enclosures. Now, the other important issue is we know, do mussels have contact with microcystis and other algae during the summer? We know that microcystis has a diel vertical migration. It can float up in the early morning to the surface if conditions are calm, and perhaps it can sink later in the day as they produce lots of carbohydrates, which acted as a ballast. We've been looking at this problem in Lake Erie from the perspective of our scums going to form. And you can see one example here. This is the actual observed concentration of microcystis. We did this with a fluoroprobe. And you can see that it varies under different conditions, and there was a mathematical model that Mark Rowe developed at our lab to show how this might be related to that. The bottom line in all this is, HABs will pop up to the surface and to be seen with the satellites on calm days. Here are some shots from a paper by Wynn et al. In Limnology and Oceanography, and you see these intense concentrations at times of high or low wind speed. In fact, if you look at the Gluro website predicting harmful algal blooms, you see that there is a defined arrows here talking about mixing likely, mixing possible, slight mixing, and then scums possible. And so whenever you have these low velocities, the stuff will float up. So there is obviously an interaction between water turbulence and whether we're going to have mussels, whether or not there's going to be a scum and at the same time no contact with the mussels on the bottom. And just going to touch on the factors affecting buoyancy. The strain of microcystis, does it have a high gas vacuole density? The colony size here shows microcystis buoyancy is the function of mean colony diameter from experiments that Nakamura did, as well as some experiments we did here at Gluro. The physiological health is driven by nutrient concentrations. Sick colonies don't have a lot of gas vacuoles. Time of day, early in the morning, there might be carbohydrate production. And you can see a whole bunch of us worked together to look at this issue, because it was important for the scum at the surface story. What we did here at GLURL is we're able to determine the size distribution of algae using a flow cam. We can generate a mean colony diameter, and then what Mark Rowe did is he taking a relationship from Nakamura, which we showed in sort of a general way, works pretty well. He defined buoyant velocity of the different microcystis. And that early slide I showed you showed that the model doesn't do too badly. However, it's not quite that simple. We've been doing experiments, actually video observations, and we showed you this in here. These are some of our data. You can see there's a huge scatter in the data. And the preliminary results from all this, the colonies from Lake Erie are generally pretty buoyant, as a lot of you know. The colonies are buoyant early in the morning. They float right up to the top. If they're from experiments we've done, if we take these algae, expose them to high surface irradiation, they'll sink during the afternoon. But if we have colonies that are not exposed to high surface irradiation, they remain buoyant. So there's a whole complex of interactions that come into play. You can see the microcystis colonies here, and you see these other elongated things. Those are Leucocyra, a diatom, and as microcystis was rising, Leucocyra was sinking. So the implications here is that under calm conditions, microcystis will float up during the late evening or early morning and be available, and not be available on the bottom of the mussels, but the big diatoms will be there for the mussels to chow down on. During the afternoon, the big colonies at the surface may sink. Under windy conditions, all phytoplankton will be available to the mussels, and what this all boils down to is, regardless of conditions, mussels are promoting microcystis dominance. So that pretty much ends what I have to say. We learned that selective rejection is an important mechanism. Nutrient excretion works in ways that are actually counterintuitive to the microcystis story. Recent work has shown that microcystis can do pretty well at low phosphorus concentrations.
All right, thank you, Hank. Again, remind folks to submit those questions in writing. And for right now, we will move on to our third and final speaker of the afternoon before we start to answer those questions. Our final presenter today is Dr. James Larson. Dr. Larson is a research fisheries biologist with the U.S. Geological Survey at the Upper Midwest Environmental Sciences Center. There he studies biogeochemistry and food web dynamics in rivers, lakes, and the nearshore zones of the North American Great Lakes. So Dr. Larson, it looks like your slides are up and you should be all set.
Okay, and I assume everyone can hear me.
Yes, we can.
Okay, fantastic. Well, thank you for having me. I'm just going to quickly go over some of the research we've been doing recently on associations between cyanobacteria and invertebrates in Lake Erie. See if I can get this. Okay, so I like to start my talks by thinking about the big picture, and the big picture for most aquatic ecosystems is that the communities that surround these ecosystems, and by this I mean the human communities, tend to want those lakes, rivers, and streams to provide a relatively simple set of things, and those include clean water and good recreational opportunities and usually some kind of aesthetic beauty. And also, at the same time, functionality for whatever commercial or industrial uses those systems are providing. Lake Erie is really no exception here. Lake Erie provides a variety of ecosystem services to the surrounding community. I probably don't have to explain all this, but there's a variety of things like clean water and recreation and industry that use the lake and are dependent on the lake. Many of the processes that support these ecosystem services are potentially impacted by cyanobacterial blooms. This talk is really going to focus just very quickly on the fisheries component here, and fisheries here refers to commercial and recreational fishing, which is a big industry, of course, in the Great Lakes in general. And in particular, I'm interested in looking at how one process in particular affects the fisheries community and is affected by cyanobacterial blooms, and that's secondary production. So for those of you who aren't familiar, secondary production really just refers to the growth of animals that can consume primary producers as food, such as eating things like algae and cyanobacteria. You can also just basically boil it down to this is the fish food. This is the production of fish food, in a sense. It's the support for the higher trophic levels that are maybe more economically important. So the overarching question here is what drives variation in secondary production? And we're looking at a lot of different hypotheses for controls over secondary production, but the ones I'm going to focus on today are those related to cyanobacteria. The first one really depends on this idea that, of course, has been mentioned already, that cyanobacteria are a really poor quality food resource for many consumers for a variety of reasons. They don't contain some of the essential nutrients that metazoans need, including including some essential lipids. And so we'd expect that because of this, places with more cyanobacteria will probably have less secondary production because they're just poor quality food. Another hypothesis here relates to the presence of cyanotoxins. Earlier talk just mentioned this a little bit, but cyanotoxins are often toxic to the cellular structure of a wide variety of animals. In Lake Erie, the cyanotoxin that's most frequently detected is microcystin, and this is, like many other cyanotoxins, harmful to a wide variety of animal life. So we'd assume that places where there's more microcystin concentration would have less secondary production as well. One thing to keep in mind here, and again, this is something that's already kind of been brought up, is that the abundance of cyanobacteria doesn't necessarily correlate super well with the abundance of, or the concentration of cyanotoxins. There are differences among different taxa and different strains and different conditions in the cyanobacteria's ability to produce a toxin and how concentrated it will become in their tissues or in the water. So here's the area of interest for this study, and in fact I'm going to narrow that down a lot. This is actually the part of the lake that I'm going to focus on. This is the western basin of Lake Erie, which is dominated by inputs from the Detroit River in the north and the Maumee River in the coming in from the southwest. And these are really different sources of water, and so this whole western basin functions a lot like a large estuarine type system where you get gradients in water quality and gradients in conditions, and it's those gradients that we're using to sort of try to understand variation in these ecological processes. So we did not actually measure secondary production directly because that's extremely difficult to do. What we did instead was put out what we're calling ecosystem process monitoring stations, which is just a really long way of saying cages and heterodendes. These are, here's what they look like in real life. This is basically a buoy attached to an anchor, which then has a cage and a heterodendy sampler attached to it. Now the cage is holding a native unionid mussel, which is a mussel that's native to the Great Lakes and most of North America. And then the heterodendy sampler, if you're not familiar, is a standardized method for measuring invertebrate communities in lakes and streams, or rivers. So what we're measuring here are basically three different surrogates for, or indices of, secondary production. the total animal mass that accumulates on the Hesterdendi as an index of how good that site is for animal growth. The size of the dreissenid mussels, because most of the mussels that are colonizing this Hesterdendi are young of year veligers that are arriving there and then growing over the course of time that the Hesterdendi is deployed. And then finally the growth of the unionid mussel that's in this cage, again as a surrogate for the conditions at the site and the suitability of conditions at the site to promote growth. These stations are deployed in late spring, and then they're deployed across a wide area of the Western Basin. This is the sites we deployed to in 2013, and then we picked them up in late summer, unfortunately a little bit before the blooms tend to end, but still it's an over summer measurement of these characteristics. Now, for this talk, I'm going to restrict it to just a small portion of the Western Basin, which is this area circled here. We have, over the last 3-4 years, collected data from 6 sites that are in this circle. Not all of them are shown here, because not all of them are sampled in 2013, which is what this map is showing. But at these 6 sites, we have these process monitoring stations, which are reporting our surrogates for secondary production. And then we also have, every other week, water quality data, which includes phytoplankton community composition, microcystin measurements, and then from NOAA, we work with Tim Wynn and get the cyanobacterial index data for this area as well. Okay, so the analysis. I just want to real quickly go over what we're going to show you, what I'm going to show you in the next few slides. Basically, we're doing a model comparison here. So we're trying to predict these response variables, that is, these indices of secondary production, using a model that either does or does not include cyanobacteria. And those responses, again, are the dreissenid mussel size, the Hesterdendi, the total mass of animals on the Hesterdendi, or the unionid mussel growth. And then the cyanobacterial data here for our first hypothesis is just the abundance of cyanobacteria, either measured as using the cyanobacterial index or measured using cyanobacterial biovolumes from count data. That's the biweekly sampling, averaged over the course of the summer. And then for the second hypothesis, we're going to actually just show you the microcystin concentrations on average, and that's measured using ELISA, which includes all the different congeners of microcystin. Okay, so I'll show you some several plots that are really the same, so I'll just go over one of them real quick here before getting into the rest of the results. The first one here, the y-axis is the response variable, the index of secondary production, and that in this case is the size of the dreissenid mussels. And then on the x-axis we have the cyanobacterial index, or the cyanobacterial data, in this case being the cyanobacterial index displayed as cell count here. And then if there's a relationship where the model with cyanobacterial data was better than the models without cyanobacterial data, I'm just putting a regression line on the plot here to show you that relationship, and also showing the R-squared value. So basically what we're saying here is that in this case, the cyanobacterial index did seem to be associated with less dreissenid mussel size. The R-squared's pretty moderate R-squared, and the effect is fairly moderate as well. If we look at our other 2 indices here of secondary production, they did not show any relationship with the cyanobacterial index. Pretty easy to see if you just look at the plots there too. This is replacing the cyanobacterial index with the cyanobacterial biovolume. This is on-site measurements. And what we see here is the dreissenid size and the Hesterdendi dry mass, accumulation of mass, are unrelated to the cyanobacterial biovolume, but there is a small negative relationship between the cyanobacteria and the unionid growth rate, which is the L-silicoidea here, Lampsilicoidea. So the conclusions for this is that I would say this is weak evidence that the cyanobacteria by themselves are negatively impacting secondary production. which is also suggested by an analysis we did that was published last year of the 2013 data, again using the cyanobacterial index. And you can kind of see this in just a map here. The mussel growth is on the upper right here, and what you see is the areas with a lot of mussel growth also happen to be areas with a lot of cyanobacteria according to the cyanobacterial index. And it could just be as simple as that, as simple as places that are good for cyanobacteria are also good for algae. And the algae themselves are really good food. Okay, so hypothesis 2 was about the cyanotoxins. Here, same kind of format for these slides. The microcystin concentration is on the x-axis. On the y-axis, we have our different surrogates for secondary production. And what we see is that in all three of these cases, we see that microcystin concentrations are negatively associated with our indices of secondary production. And if you look at the R² values, there's more variation being explained in the dreissenid size, and the effect size is a little higher with the dreissenid size too, although I haven't shown effect size here. In all three of these cases, these aren't actually linear relationships, however. These are log transformed data. So what we're seeing is a log linear relationship between these two variables. So for the second hypothesis, there's much stronger evidence that the microcystin are negatively affecting these invertebrate consumers' overall secondary production. This is a little bit a touch surprising, perhaps. I mean, yes, cyanotoxins are called toxins because they're bad for most animals, but bivalves are consistently referred to as very resilient or resistant to cyanotoxins. I've read I don't know how many papers about exposures to cyanotoxin to look at accumulation of toxin in muscle tissues, and it's always got a line in there in the results that's like, oh, we doused these things with cyanotoxin and nothing died. So it's not entirely obvious that these should have a big effect on secondary production. But there are a lot of sublethal effects, and those are being increasingly well documented, as you know, some of the talks earlier in this session have shown. So this maybe isn't a huge surprise. Dreissenids are probably the most, the source of most secondary production in the western basin of Lake Erie. There's been a few studies that have measured secondary production directly, and dreissenids seem to make up the majority and even a vast majority at times of secondary production in the western basin. So it's likely that the biggest effects we saw were on the dreissenids themselves. It's likely this indicates this is having a negative impact on the system, on the secondary production in the system. And then to put this into a little bit of context, the 3 years we sampled, this is the bloom severity. Well, this is a slide from the forecast, but it shows the bloom severity that NOAA quantifies for the last 15 or so years. And here in this box are the years we actually are sampling from for this talk that we sampled during. And you can see these weren't good years. These were pretty severe cyanobacterial conditions during those years, just generally in the western basin. Of course, at our particular sites, they were a little different. But what we overall conclude is that during this time period, there's certainly places where secondary production seems to have been negatively impacted by the presence of these cyanobacterial blooms, and in particular the presence of microcystin as a toxin. And likely this is going to affect the higher trophic levels that are supported by these secondary producers. So that's all I've got for today. Thanks again for having me on. And if you had questions you wanted to send me directly, email's right here. Otherwise, if we have time, I'll answer questions.
Thanks, James. So that does conclude our presentations for today, and so we're going to move on to answering your questions and respond to any comments that you might have. As a final reminder, you may type in any questions you have into the questions pane of the GoToWebinar dashboard. So we have received a few questions as we've moved along, so we will ask those now to our speakers for today.
So our first question is for Donna. Joe Duras asked, do you plan to test mixtures of cultures, since cyanobacteria occur in mixtures in the environment?
So at this time, we are isolating individual species and not doing mixtures because we don't even know the mechanism within the individual species. So we actually want to go the opposite direction and try and find what within a specific cyanobacteria. If we are able to identify a compound within a particular bacteria, within a cyanobacteria, then we can start looking at going bigger picture. But normally I'm a broader researcher, but at this point we need to know what's doing it, what's causing it. So no, we're going to focus down, not up right now.
Thank you, Donna. And for all of the question askers, if you want to ask a follow-up question, once you hear a response, feel free to type that into the questions panel as well.
Our next question is for Dr. Larson. And the question is, do you think that induced spawning during the warm water summer season would adversely affect juvenile and adult health?
Perhaps. Yeah, I mean, I don't think they do very well in the warm conditions. Maybe somebody else on the team has more insight into the mussel biology, though.
I was going to say, that seems like a question that could also be addressed to Donna.
So can you restate it?
This question was, do you think that induced spawning during the warm water summer season would adversely affect juvenile and adult health? So I think this question was kind of bringing together information from your talk on algal abundance potentially inducing spawning with the finding from James's talk that mussels in high algal areas had lower average biomass.
I do suspect that anything that affects an organism's metabolism is going to have some sort of cost effect. So I do think that it is going to have an impact. I don't know if it's enough of an impact. It may make them more vulnerable to something else. I don't think it's going to be enough that it's going to shut down or something like that. But I do think it could potentially impact secondary production because I do think there's going to be a cost anytime you spawn. And if you're doing it repeatedly and more often, that is a lot of calories. That is a lot of energy being expended.
Thank you, Donna and James. The next question was also addressed toward, I think this is another one that could be opened up to any of the panelists. Here at Crane Lake in central New York, we have microcystis blooms that have become more pronounced since zebra mussels came in during the early 2000s. We also had grass carp introduced in the late 1980s. The grass carp are now almost gone, and we have more prolific rooted aquatics, to the point that some folks want to reintroduce grass carp. What would be the impact of increased short-term nutrient cycling from grass carp on the mussel microcystis synergy? I think that's when we can definitely open up to any of the panelists. James, you want the first crack?
Yeah, no, I want to hand it off to the NOAA people.
Yeah, so the issue is with the grass carp were there, the mussels came in, you got the harmful algal bloom, and then at the same time the grass carp disappeared, then you got more weeds. Well, the dreissenids are maybe have an effect on the water clarity, which would have an effect on the vegetation, various macrophytes and benthic plants. The microcystis blooms are totally consistent with that. Depending on the nutrient concentrations in the lake, if you got mussels down near the bottom, they're clearing the water in the shallows and excreting nutrients which the benthic plants would like very, very much. Now, when the grass carp I don't know if they root around the vegetation and cloudy up the water in addition to consuming the plants, but there might be a factor there as well, I'm guessing.
Yeah, so this is James again. In my experience talking to people who worked with grass carp more, it is the inorganic sort of turbulence they create that can be a problem for the plankton producers. So I don't know how the nutrient release they might facilitate would influence that either.
Sounds like there are some open questions there.
Donna, is there anything you wanted to add to that discussion?
No, I think they covered it fairly well.
All right, so we'll move on to the next question, which we will also pose to all three of our panelists. Has anyone looked at the effect of cyanobacteria on veliger survival or veliger feeding on these types of algae?
Yeah, this is Hank. A few comments on that. We've reared veligers in the laboratory, and there would be a couple of issues involved. First of all, microcystis would the colonial form would be too large to ingest, and the other thing, if there were individual small colonies, which would be individual cells on the order of 3 to 6 micrometers in size, they would be of poor nutritional quality. They lack essential polyunsaturated fatty acids. So one, they're too big, and any cyanobacteria if they're small enough to be ingested, are poor quality food. So in general, I would say summer with lots of microcystis or any cyanophytes is a bad news for the veligers.
Donna or James, did you want to add anything to that?
Working with veligers is incredibly difficult. We recently did, working with the USGS, develop an assay using veligers, and we looked at a lot of toxins. And one of them that we were just doing on the side was indeed using a microcystis strain from Texas that wasn't colonial. And we were putting it in there, and like Hank said, they were not doing well. And so we would mix it with something more palatable for the veligers. And in general, they still did not do very well, but we didn't really follow it. It was a small project we were doing alongside looking at contaminants like fire retardant. So I suspect it is going to have a negative effect. One, like Hank said, starvation, but I do suspect there's another effect going on too.
The next question was also addressed to everyone. What are the potential differences between zebra and quagga mussels, and hypothetical differences in the species' relationship to cyanobacteria? Has anyone looked at the differences between those two?
Yeah, this is Hank. Huijian Tang, Chinese visitor who worked for me, did most of her work with quagga mussels, and she did an extensive review in terms of comparison. And in terms of what they feed on, is pretty much the same story. I think with the quagga mussel, the issue would be they'd be able to go into deeper waters and have a larger impact on a lake system.
Again, Donna or James, do you want to add anything?
No.
No.
All right, well, the last question we have right now is directed at James, but Donna or Hank, if you have something to add, please feel free to jump in. Does correlation between cyanobacteria and secondary production mean that cyanobacteria is the cause?
Well, no, of course not. It's a statistical association. We certainly have lots of reasons to believe there would be negative associations between cyanobacteria and secondary production, just because, as Hank mentioned, they're poor quality food. I mean, not just for dreissenids and not just for bivalves, for basically everything. There's been lots of studies on zooplankton, and they also tend to not do well when they're feeding on cyanobacteria. So it's not that the association means there's a relationship there, but we have lots of theoretical and conceptual reasons to think there should be a negative association as well.
Thanks, James. Right now, we don't have any other questions submitted, but we'll give folks another minute to submit them if anybody has any they haven't typed in yet.
And while we're waiting for that, we've heard 3 really interesting talks today on different aspects of ways in which chemicals from probably not the microcystin, but other chemicals associated with cyanobacteria are affecting the specific physiology and reproductive potential of mussels. That has implications both for what happens in the lake, especially as the seasonality of the blooms shifts, but also has implications for technology that could be used to control mussels. We heard on over 2 decades of research on the mechanisms of dreissenid mussels potentially facilitating the blooms of microcystis through selected feeding, selected rejection, through nutrient release, through the vertical distribution of them, of the microcystin, the microcystis. And we heard on how in the lake the distributions of secondary production relate to, especially of the dreissenid mussels, relate to the cyanos and even more strongly relate to the microcystin toxin concentration in the water. Do any of our speakers have thoughts on connections between those issues? Are there things that you heard today that will impact how you're doing your research, or other global take-home messages from these 3 talks that you would like to share?
Well, I do think, this is Donna, that expanding upon some of these research questions, like Joe Duras pointed out, looking at combinations of cyanobacteria, how does that differ than single species? Looking at these, a lot of the questions are what tied these things together. Taking it the next step after reproduction, velagers, what are the impact? So there's still so much to be done, and looking at productivity, secondary production, just so much work to be done that I think just identified by these 3 on how little we really know about the physiology and understanding their interactions. But at the same time, we know a lot about kind of those ecosystem level questions and how it affects different phytoplankton species. But we're still nowhere near having all the answers. I think these 3 talks actually highlighted that with the questions coupled with them.
James or Hank, any thoughts or reactions?
Yeah, well, one thing, this is Hank, one thing I'm interested in these days is, we've talked about microcystis, there are other bloom producers, Aphanosomnon and Anabina, and there may be some, this is something we're planning for the near future with my colleagues here at Goodall, Tim Davis, as well as Ashley Elgin, is what are interactions with the other HABs? The nutrient excretion may affect, some of the nitrogen-fixing blue-greens, Phanosomenon and Anabina. From what little work we've done, it may be that the mussels may prevent a shift from Microcystis to Anabina by scarfing up the Anabina and at the same time excreting phosphorus, which could be used by Microcystis to keep it shifting to phosphorus limitation. So there are all sorts of nuanced interactions there that might be explored.
This is James. I mean, I think the thing that these talks together make me think of is that it's very interesting how the feedbacks between these two things are likely to continue to perpetuate them. If you have this large population of mussels out there, they're going to continue to facilitate Microcystis invasion. And the process that you might imagine would reduce that would be the toxicity of those cyanos themselves, and as they wipe out everything else, they might, or outcompete everything else, they might reduce the viability of the mussels, but that doesn't seem to totally happen. So it seems like this is a whole state shift.
Thanks for those thoughts. We did receive one more question, which we'll ask before we wrap up for today. And so can any of the panelists speak more about the nutrient stoichiometry and how changes in the nitrogen-phosphorus ratio may impact toxin production in invasive species?
Yeah, that's an interesting question. One of the limiting nutrients for microcystin production is nitrogen. If you don't have enough nitrogen in the system, the capacity to develop microcystin is lower. So the mussels are excreting nitrogen, but more particularly they're excreting ammonia, which Microcystis relative to, let's say, diatoms is pretty good at sucking up. So there may be shifts in these nutrient ratios which could favor toxicity. And there's the whole thing is pretty complicated. It depends on, of course, the stoichiometry of your system. It depends how the mussels are feeding. If they're not feeding, they'll be pumping out nitrogen, but no phosphorus. So it's a very interesting story, and we've just begun to scratch the surface on that. And perhaps I'll send something we have in press to add to the workshop to this site where we discuss that problem at some length.
Thanks, Hank. So we are going to wrap up for today. We want to thank all of our participants. Obviously, this is a very rich subject, and so we encourage your follow-up questions or comments to either collaborative about this issue. As a reminder, the webinar recording will be posted on both the HABs Collaboratory and the Invasive Mussel Collaborative website within the next couple of days. And again, please feel free to contact us with any follow-up questions or comments. Thank you to all of our panelists for their very informative presentations, and thank you again to all of the participants. That concludes our webinar for today.